So we are saying that the central vacuole and the cell wall are not found in animal cell. But you see the others are the same. You can see the nucleus here. You can see the nucleus here, the nucleolus, nuclear pore. You see these pores. And then chromatin, these white things. Nuclear envelope, just this envelope. So found in the nucleus. You will see the Golgi apparatus. All these things are there. Mitochondrion, this sac here. But we have something new that is not present in animal cell. That is chloroplast. So this one not present in animal cell. So just make sure that you study, you memorize all the parts of these cells. Now let's look at all these parts. Also be able to differentiate between a plant cell and an animal cell. Now let's look at the nucleus itself. What is the function of the nucleus? So the nucleus contains the genetic material. So the DNA of a cell is found in the nucleus. The cell has a fluid that we call nucleoplasm. So it's semi-fluid. This plasm is found within the nucleus. Then it has this chromatin. These chromatin are like thread-like, like kind of DNA. They are grainy in appearance. You see, they look like white grain. Right? They are thread-like DNA. So they are the DNA that the nucleus contains. The DNA is found within the nucleus. Then we have the nucleolus. Nucleolus is just a dark region of chromatin, and the nucleolus produces RNA. Chromatin has DNA, right? So let's take note of that. Chromatin has DNA, while the nucleolus has RNA. So this one has RNA. So when you think about the synthesis of protein, you can easily say that the synthesis of protein will occur in nucleolus, because that's where the RNA is found. And RNA composes ribosomes. Ribosomes are the place for protein synthesis, the place for making protein. So for making protein. So if you want protein, just go ask ribosomes. They will make some for you. Then the nuclear membrane, it's a double layered. It surrounds the nucleus and has these large pores. So the membrane is double layered, surrounds the nucleus, and it has a larger pores. So what we are doing is we are describing each and every part of the cell, and also thinking about its function. So now if you look at the cell, this is the membrane. This whole thing is membrane. These are the pores that we are talking about, the large pores. This is the lipid bilayer. This part is polar. Inside here is non-polar. So when we go back to the nucleus, chromatin is the whole thing here. while nucleolus is this thing inside. So I'm guessing this whole thing is chromatin. If we go back here, I said chromatin are these, but no, they are not. This whole thing inside here is a chromatin. So the membrane, because it's bilayer, it has inner membrane and outer membrane. Now, what are ribosomes? As we said, ribosomes are a site for protein assembly. Ribosomes are just RNA subunits. So they are composed of RNA subunits. If you want to make protein, you need RNA. So these can be found as just free ribosomes, not bound to anything, or can be bound to endoplasmic reticulum. So they are either bound or not. So really, we have another important term, an endomembrane system. So this endomembrane system includes the nuclear membrane. So if I ask you what is the endomembrane system, just tell me it includes the nuclear membrane, endoplasmic reticulum, Golgi apparatus, and the vesicles. And we'll talk about these others like the Golgi apparatus and the vesicles and see what they do. So it includes four things. Endomembrane system includes nuclear membrane, endoplasmic reticulum, Golgi apparatus, and the vesicles. So remember those four things that make up the endomembrane system. So as we checked, as we saw last time, endoplasmic reticulum, we have two types. We have rough and smooth. So this rough endoplasmic reticulum is just a system, complex system of those channels we saw, Gadimizere, right? So we saw them like this. All those channels, they are complex system of membranous channels and circulars. Forget about circulars, just channels. So these channels have ribosomes on them, right? So this is ribosomes attached to the endoplasmic reticulum, while other ribosomes are not attached to this membrane. So this is where you see protein is made, protein is assembled. So protein is assembled in the ribosomes. These ribosomes are found in the endoplasmic reticulum. Once these proteins are made, are assembled, they will go through the channels, right? We are saying each one is a channel. Once the proteins are made, they will go through the channels and they go into the channels to be processed. So what does processing means? Once you make the proteins, you say, now I've made you, go into these channels. The channels of the endoplasmic, of the rough endoplasmic reticulum. Once they go there, sugars will be added. So addition of sugar chains to form glycoproteins. So glycoproteins are proteins with sugar part. So I just say proteins with sugar part, because that is coming from glycogen. So the sugars are added in the endoplasmic reticulum, or in these channels that we are talking about here. Then after that, the protein is released in vesicles. We'll see what the vesicles are. The vesicles are just like a vehicle that carries protein. So this happens in the rough endoplasmic reticulum. So if I ask you, what happens in rough endoplasmic reticulum? One, you tell me it is a site of assembly of proteins. Two, the assembled proteins will enter channel, the endoplasmic reticulum channel, for processing, where sugar is added. Three, the proteins will be released in vesicles. So this is a rough endoplasmic reticulum. What about the smooth endoplasmic reticulum? This part synthesizes lipid products. So the phospholipid that are found in the bilayer are synthesized by the smooth endoplasmic reticulum. It also synthesizes steroids. So if you ask me, what is the difference between rough and smooth endoplasmic reticulum? The rough assembles protein. The rough endoplasmic reticulum will assemble proteins, while the smooth endoplasmic reticulum synthesizes lipids, lipid products such as phospholipids and steroids. Then these ones are also released in vesicles. So in both cases, what is produced in rough endoplasmic reticulum and smooth endoplasmic reticulum is released in vesicles. So this is what we are talking about. Rough endoplasmic reticulum is all these channels you see here, while smooth endoplasmic reticulum is this side you see here. And you see what? This is a vesicle, a carrier that leaves this endoplasmic reticulum chain. So the whole thing here is like a channel, this channel. And we said that the sugars are added in the channels. You can see these are sugars here, carbohydrates. These are sugars that are added to that protein. And then we transport them from here. We transport the proteins through vesicles. So we transport them from channels through vesicles. And this is a vesicle. So what is the vesicle's job? Is to transport protein, to transport lipids within the cell. So that's a better diagram of a cell. You can see here the nucleolus. Chromatin is the whole thing here. Nuclear envelope is this one covering the nucleus part, and these ones are the rough endoplasmic reticulum, the channels that we are talking about. Then we have a Golgi apparatus. What does it do? Now, we say the endoplasmic reticulum, the rough endoplasmic reticulum assembles protein with some sugars. The smooth endoplasmic reticulum assembles fats, lipids. And then after the assembly, the rough endoplasmic reticulum and the smooth endoplasmic reticulum will put all those lipids assembled and protein assembled. It will put them in vesicles. Now the vesicle will transfer to the Golgi apparatus. The Golgi apparatus will receive the protein and the lipid from the vesicles. Then it will package them, process them, and distribute them. So Golgi apparatus distributes protein and lipids in the So this is done after packaging them. So the vesicles are how this is done. The vesicles will fuse with the Golgi and release the product inside. So, and we'll see what the Golgi is. So like let's say the Golgi is here, the vesicles are coming here, the vesicle will go all the way and fuse with the Golgi here, and then release whatever is inside in the Golgi apparatus of the cell. When the Golgi gets all that, then the Golgi will distribute whatever was in the package. Will distribute the protein and the lipids. And also within the Golgi, the glycoproteins will be modified. So really, Golgi apparatus does different things. First, it will receive protein and lipid vesicles and then process and distribute them. 2, it will modify the glycogen proteins that came through the vesicles. And then afterwards, they are put again into other vesicles that we call secretory vesicles, and then released to the cell membrane. Definitely the cell membrane will say, do I need to put you out of the cell, or do I need to keep you? So Golgi apparatus also produces what we call lysosomes protein, a lysosome protein that contains vesicles within cells. So this is a protein that we call lysosome. This protein is formed by the Golgi apparatus, and we will see from the diagram what lysosome is. Now this is the diagram. This is the Golgi apparatus. These ones are the vesicles. You see the vesicles are coming in. When they come here, you see this vesicle fusing and then releasing whatever contents it had. What? These are coming from endoplasmic reticulum to the Golgi apparatus. Once all this is done, then the Golgi apparatus will release secretory vesicles. These ones are coming in, they fuse with this, and these ones are being released. Now, this one goes all the way to the cell membrane. You see, here is the cell membrane. Now it releases whatever contents it has through the cell membrane. So what are the lysosomes? The lysosomes are found inside here, inside these vesicles. See? They are just a protein that we call lysosome. Its job is to digest molecules or all the cell parts. That's what the lysosome does. It is an enzyme. But where is this lysosome produced? It is produced in the Golgi apparatus. It is produced here. Lysosome is produced here. You can see these are secretory vesicles. These ones are transport vesicles. Transport vesicles carry protein and lipids from endoplasmic reticulum, while secretory vesicles carry proteins, maybe, and lipids from the Golgi apparatus to the outside of the cell. So take note of all these, because this is kind of anatomy. You need to memorize all these. What are they? Where are they found? You need to memorize these diagrams and all the parts of the diagram. So the lysosome, they contain a hydrolytic enzyme. We know that hydrolysis is breaking things apart. So we know that hydrolysis is breaking things apart. So the lysosome contain hydrolytic enzymes and are involved in intracellular digestion. Digestion within the cell, right? So these lysosomes work inside the cell, not outside. They will break down things. They'll break down things in the cell because they are an enzyme. So normally they will fuse with the cell membrane. So the cell membrane has these macromolecules, protein, lipids, fat. So this lysosome will fuse with the cell membrane, knowing that this cell membrane has macromolecules. Once it fuses, it will digest those macromolecules into smaller molecules. For example, it will digest starch to glucose, or glycogen to glucose. But also lysosomes, may also help with cell death, which we call suicide sacs. So the cell that is about to die, the death is quickened by the enzyme lysosome. So how do they do that? They rupture old cells, they rupture them and use the enzyme to digest the cell. So the lysosomes are important. When they see that this is an old cell, they will get into contact with it, rupture it, and then digest it. Then the cell has energy-related organelles. So what are these energy-related organelles? We have chloroplast. A chloroplast in a cell is a site for photosynthesis. We know the green part of a cell is a chloroplast. So a chloroplast will produce food in plant cells. So how does it produce food? It interacts with energy from the sun and then carbon dioxide from the air to make glucose. And release energy and oxygen. So it makes, actually not release energy. It uses energy it absorbs energy from the sun and combine it with carbon dioxide and water to form glucose. So that process, formation of glucose, starts here. Right? Through photosynthesis. So all the starch that is made by the plants starts with glucose. And this glucose is made through the process of photosynthesis. And where does that occur? It occurs in chloroplast. So this is what it does. It captures energy and converts it to chemical energy in form of food macromolecules. So the chloroplast is surrounded. The chloroplast is surrounded by double outer membrane. And the membrane system of chloroplast is known as thylakoids. So just the name. And there are weird names here. If you put the thylakoids together, they form a grana. So let me start again. The chloroplasts are found in a double outer membrane surrounding. We have in a chloroplast, we have double outer membrane surrounding a fluid-filled stroma. So the stroma is inside. That stroma is surrounded by double membrane, outer membrane. And the membrane system of chloroplast is thylakoids. And if you stack so many thylakoids together, they form grana. So these are thylakoids. Thylakoids, you stack them together, they form this grana. You see inner membrane, outer membrane. All this thing is chloroplast, the whole thing here. So if you say, what are the parts of the chloroplast? You say the parts of the chloroplast are thylakoids, stroma, and grana. Double membrane here. Those are the parts of chloroplast. Chloroplast is where the chlorophyll is found. So this is where chlorophyll is found, in the chloroplast. Then the cell has mitochondria. This one is also related to energy. Chloroplast captures energy from the sun. Mitochondria will produce energy in animal and plant cells. Surely through respiration. Right? So both plants and animal undergo respiration and release oxygen. This is done in the mitochondria. Mitochondria is a site of aerobic. So aerobic respiration is oxygen respiration. Respiration where oxygen is present. That's aerobic respiration. This happens in mitochondria. And the result is the production of adenosine triphosphate. This is the energy molecule. So ATP is an energy molecule. that is produced through aerobic respiration or just through normal respiration. The mitochondria is also has an outer double membrane that surrounds the fluid-filled matrix inside it. So mitochondria also has fluid inside it and is surrounded by double membrane like chloroplast. So inside we have folded membrane. Inside the mitochondria we have folded membrane that we call cristae. Now because of folding, like if you fold them like that, we increase the surface area. You can see all the surface area here because of this folding. So cristae will provide increased surface area for the production of ATP, because the ATP will need to be in contact, or the chemicals will need to be in good contact to produce ATP. When you increase surface area, you are increasing the points of contact for a good chemical reaction. That's why cristae are important. They provide that increased surface for ATP production. So, how does the mitochondria look like? You see this fluid? The fluid is matrix, like fluid filled matrix. So this is the double membrane, the outer membrane here, the inner membrane here, and you can see the folding itself. The folding, this is what we call the cristae, this folding, and this folding increases the surface area for the production of ATP. So there is an interrelationship between mitochondria and chloroplasts. What do they do? Chloroplasts will convert light energy to chemical energy in form of organic food molecules. And then this energy can be used by all organisms in cell respiration to make ATP. So first, chloroplasts will convert light energy into food, and then when you eat the food, the food will undergo respiration to form adenosine triphosphate. So these two are energy molecules. Where are chloroplasts found? They are found only in plants and algae. So animals do not have chloroplasts because animals do not make food. So I would say animals do not have chloroplasts. Do not have chloroplasts because animals do not make their own food. That's what is happening. Animals do not make their own food, therefore they do not have chloroplasts. So when you talk about photosynthesis, the reaction that happens in chloroplasts, we have light energy added to carbon dioxide and water to produce carbohydrate and oxygen. Then, once you produce carbohydrates, then you have carbohydrates will react with oxygen to form carbon dioxide, water, and energy. The energy is in the form of ATP, adenosine triphosphate. So what does it mean? Whatever you require in cell respiration is produced in photosynthesis. Whatever you require in photosynthesis is produced in cell respiration. So let me stop here and continue later on.